very much for your invitation. Hello, everyone. My talk is articulated pharmacological properties of complex types and concept cells. Each year, thousands of papers were published in the world evaluated anti-cancer drugs, but only a very small number of molecules reach patients. Bridging the gap between lab research and clinical reality is a major priority for biomedical research. Drug design and synthesis methods are essential steps in creating new therapeutic agents. Medicinal plants remain an essential and relevant source of inspiration for these developments. Let me share some examples of molecules or chemotherapy derived from medicinal plants. First example, interesting and progressing. This is a alkaloid derived from the Madagascar periwinkle and is used for the treatment of leukemia, lymphoma, testicular, and breast cancer. Second example, paclitaxel, derived from the bark, the cox bark, or the Pacific fuel tree, is used for the treatment of breast cancer, ovarian, and cancer. Camptothecin is derived from Campotica tree and used for colorectal cancer and ovarian cancer. Next example, glucoside, an alkaloid target topoisomerase 2, is derived from a ripe plant and used for the treatment of testicular, small cell lung, and some leukemias. The current approach used by pharmaceutical industry include drug design, cell culture, animal testing, and clinical trials. The use of cell lines to test anti-cancer drugs starts in the 1950s. Paper published Nature in 2022 estimated the number of human cancer cell types 947. These cancer cell lines covered all the types of cancers. Cell lines is very easy to use, have an easy manipulation, cost effectiveness, and high reproducibility. But the use of these cell lines is considered one of the reasons for the failure in detecting clinical responses. Because first, that is of 3D structure, or simplified condition, limited cell and cell matrix interaction, and the absence of cell heterogeneity. Animal models is historically used before the cell lines. In fact, for example, cisplatin is used only for preclinical research, is used or tested only with animal models. In general, animal models have a 3D structure, a short gestation period, an easy genetic manipulation, and the communication between different organs. We can make patient-derived tumors evident. However, ethical issue, high costs, difference between human and neural physiology, limited reproducibility, and essentially low predictive values, and the lack of concordance with clinical trial is a must for to research another alternative like 3D cellular model that could be a greater alternative. 3D models mimic cell polarity, shape, nutrient and oxygen gradient, as well as interactions between cells and surrounding stroma. An increasing number of preclinical research studies are incorporated 3D models such as spheroids and organoids. For example, this work is an Egyptian team. The scientists selected 500 medicinal plants and screening these in RCT 116 2D lines. After the first selection or screening, 41 plants demonstrated over several cancer cytotoxicity at 100 microgram per milliliter. These 41 extracts were then tested on spheroids using first RCT 116 cells and spheroids derived from a normal epithelial cells. The results suggest 3 plant extracts were effective in both tumors and the models. Next work, for this work is breast cancer. The first part is a classical method of selection with a screening with MCR7 cell lines in 2D models. After researcher and scientists has analyzed the impact of this plant extract on the size and the morphology of spheroids. It's a very easy technique to make the models. And staining the apoptotic cells with final assays. Just for to explain, we can make 2D and 3D model in the same paper. Next one is the green tea, for this work, researchers have evaluated anti-cancer potential of this stack with the type and the size and the morphology of different spheroids developed with MCF7 cells. Next work is to analyze the combination between doxorubicin and the chemotherapy and 2 plant extracts. The first part of the work is to analyze in 2D models in leukemia, 2 leukemia cell lines, and 2 normal cells, mesenchymal cells and endothelial cells. This is important, TC50 have a maximum inhibition concentration values were calculated, and then with confocal microscopy, we evaluated the cytotoxicity of 2 extracts and doxorubicin. It's very interesting work because here we can see that this extract is not toxic for mesenchymal and endothelial cells, but it's toxic for these leukemia cell line. For analyzing cell death, in green is live cells, and the red is dye for dead cells. But the difference between these cell lines is difference are observed depending on the spheroid types. Here, for example, we can see with spheroid used normal cells, we don't see cytotoxicity, but we see a cytotoxicity with leukemia cells. For chemotherapy, we can see a cytotoxicity for normal cells and leukemia cells. Next work is very interesting work. The scientists for this work developed an environments and after injected them subcutaneously into mice or to make an in vivo study with organoids. And then we can treat the mice with the plant extract. This, for example, the tumor prevented from mice. This work is published recently. Scientists used isolated cells from patients, created an organoid with the cells, and here we say the effect of plant extract on the shape, on the size of the spheroids, and with polygon iodine, you can see the cytotoxic effect of extracts. This is my last work in the field of cancer therapy. With hospital Aliza Atmena, we analyze the cytotoxic effect of leukemia cells or artemisia complexes on leukemia cells. We isolated cells from patient, and then this is 15 cells from patient, and then we make a 2D cell culture of patient cells, and it's new study. After we analyze the apoptotic effect, the effect of anticytes on cell cycle, the mechanistic effect is the apoptotic effect on the mobilization of mitochondria. And here we see the effect of artemisia complexes and the number of cancer stem cells. We see that for some patients, we can reduce the number of cancer systems with blood extract and with this chemotherapy, C. Thank you very much.
Thank you very much, Mohamed. Quick question. Should we drink more tea? Green tea, then black tea? Green tea? Just as just a funny question. So anyone has a question in the room? Yes? Oh, yes. Oh, sorry. Our friend.
Thank you for your presentation. Just, I want to know, what are the pharmacological properties of medicinal plants?
Like the anti-cancer potential of plant extracts.
Thank you. Thank you for the presentation. I would like to know the plant extract. By the way, I come from a plant biotechnology background, so I would like to know the extract. It will be used like to create drugs, or what will it be about in the treatment?
For this work, it's a ethanol extract. We don't have isolated molecules from this one. But we have characterized the composition of artemisia complexes.
We add the extract to the cell culture?
Yeah. A different concentration.